Hi, I'm Naofumi Yoshida. I'm a PhD student at Riken Kobe in Japan. In this presentation, I'm going to talk about my research, which asks the question, what features of cortical microstructure does diffusion MRI reveal? Specifically, what histological features does the recent diffusion neurites model correlate with? To answer these questions, I'll present the research results that we found by comparing diffusion MRI-based neurite with histology data in multiple primate brains. First, let me introduce the background of my study. Recent methodological advances in MRI, particularly in Human Connectome Project (HCP), allowed us to investigate in vivo cortical organization in humans in great detail. We have developed acquisition and analysis pipelines for non-human primate (NHP) that adapt HCP protocols and enable a fair comparative study across living primates. Indeed, T1 weighted by T2 weighted ratio mapping provided valuable insights into consistencies and differences in cortical microstructure across primate species. Diffusion MRI provides an alternative way to explore cortical microstructure. One of the recent trends in diffusion MRI technique is biophysical model-based strategy to estimate brain microstructure, such as NODI, which estimates the density and orientation coherence of neurites at each voxel, here referred to as neurite density index (NDI) and orientation dispersion index (ODI). In our previous study, we applied the NODI model to HCP dataset, developed surface mapping pipeline, and found that the spatial distribution of NDI was quite similar to T1-T2 myelin. Regional neurite density was moderately correlated with T1-T2 myelin. So, we considered NDI may partly reflect the restricted diffusion of myelinated fiber. However, much less is known about the cellular organization estimated by NODI, because it's impractical to directly measure neurite density in histology. To address this issue, we hypothesized neurite density index (NDI) correlate with neuron density because we thought the neuronal membrane would also restrict water diffusion, and there must be some correlation between the amount of neurites and neurons. As for histology data, Herculano Fuzel accumulates the cortical neuron density dataset in many mammalian species using a simplified but reliable quantification method, isotropic fractionator, or IF. So, our aims of this study is to investigate whether NDI covariates with neuron density across cortical areas and primate species. We correlate diffusion-based NDI with IF-based neuron density. The second aim is to investigate variation of cortical NDI maps across primates to understand the homology across species. We acquired 18 rhesus monkeys, 12 cynomolgus monkeys, and 9 marmosets using a 3 Tesla MRI scanner and custom-made receive coils. For human, we used 505 subjects using HCP data. For macaque and marmoset, we applied acquisition parameters harmonized to the HCP acquisition. All datasets were preprocessed by using HCP-NHP pipeline, applying the distortion correction, registrating to the structure space, fitting NODI model to the data, and obtaining the NDA map on the cortical surface. Then, we compared these NODI maps with histology. First, we used macaque cortical neuron density data by Collins in 2010, mapped the data onto our average cortical surface, and compared it with the macaque average NDI map. Second, we investigated the whole cortical average values of NDI for each species, and compared them with mean cortical neuron density calculated from published dataset by Herculano and Fuzel after confirming the validity of cortical mass. Third, we also verified cortical orientation dispersion index, ODI, by comparing with cortical myelin fiber complexity in Gallius staining in macaque brain. Finally, we investigate spatial patterns of cortical NDI mapping across species. Here's the result of comparing cortical NDI with neuron density across cortical areas in macaque. The left panel shows the original maps of Colin's macaque cortical neuron density, which is mapped onto our macaque average surface map. The same passation was applied to the 30 macaque average NDI map, and we found similar cortical distribution, excluding anterior temporal area, which had high susceptibility artifacts. A strong correlation was found between cortical NDI and neuron density across cortical areas, with a correlation coefficient over 0.9. As for cortical average of NDI in 4 primate species, shown in the left and middle panel, it is interesting that humans have a minimum NDI value among primates, followed by cynomolgus, rhesus monkey, and marmosets, suggesting that smaller primates tend to have overall higher cortical neurite density. Interestingly, this pattern was again found in neuron density. A plot of the 4 cortical average of NDI against neuron density showed a positive correlation, although the number of the species is limited for availability of the neuron density data. That said, this principle of cortical mass and NDI is consistent with the previous literature, showing the negative correlation between cortical mass and neuron density in a greater number of primates. Next, with regard to macaque cortical ODI, we manually registered diffusion image to myelin staining, analyzed mean ODI and circular direction variance in 4 regions of interest. We found ODI and circular variance had a positive correlation. Despite the small number of rows, ODI may reflect the complexity of cortical myelin fibers, which is consistent with the previous study published by Gruss et al. Here's species homology of cortical neurite density. In this analysis, cynomolgus and rhesus monkeys were averaged into a single macaque surface map, and the color codes of signal intensities were adjusted in each of 3 species to facilitate identifying the differences across areas. As you can see, the spatial distribution of NDI was similar across species. NDI high in primary motor and somatosensory cortex, auditory cortex, MT, visual cortex, cingulate gyrus. NDI low in frontal, parietal, and temporal association areas. Past histological study in macaques showed cortex neurites and neurons are sparsely distributed in prefrontal and inferior temporal regions, and densely distributed in primary visual cortex. Marmoset cortical neuron density map by others is also consistent with our NDI map. Interestingly, these low NDI association areas disproportionately expanded in the human brain, which may be related to evolution of high-order cognition. Our data give us an additional insight that sparseness of neurons and neurites may facilitate dendritic arborization, synaptic reorganization, and neural plasticity. In summary, we found that cortical NDI was strongly correlated with neuron density across cortical areas and across 4 species in the living brain. We also found that cortical NDI showed similar spatial distribution across 4 primates. NDI was high in the primary regions and low in the association areas. These findings suggest that NDI may have potential for examining in vivo cortical cellular development and degeneration. This concludes my presentation. Thank you for your kind attention.